From ARUP Laboratories on the campus of the University of Utah, welcome to the LabMind podcast. I'm your host, Dr. Brian Jackson.
Today is Thursday, the 2nd of May, 2024. Today's LabMind interview is about pharmacogenomics and what makes this an important domain within laboratory testing, what makes it a little bit different from other areas. And I'm hoping we can explore some of the reasons why it has taken longer to take off than some have expected it to. Our guest today is Dr. Yuan Ji. Dr. Ji is Section Chief of Molecular Genetics and Genomics at ARUP Laboratories. She is also Associate Professor of Pathology at the University of Utah. Dr. Ji received her PhD in molecular pharmacology and experimental therapeutics at the Mayo Clinic, followed by fellowships at Mayo in pharmacogenomics, clinical molecular genetics, and clinical pharmacology. And she also recently completed her master's degree in business administration here at the University of Utah. Yuan Ji, welcome to LabMind.
Nice to be here.
So maybe we should start this with a definition. Hopefully the listeners know what pharmacogenomics is, but if someone asks you what that word means, how do you define pharmacogenomics?
Pharmacogenomics is the study or discipline to characterize the inheritance. And nowadays, it also includes the acquired mutation changes in our DNA profiles on the drug response phenotypes. There's a large variation of drug response phenotypes among the individuals. So pharmacogenomics is the marriage between pharmacology and molecular genetics. And it's a newer discipline.
What was it that motivated you to do a fellowship in pharmacogenomics?
I was a graduate student in my first year. We were asked to choose a discipline or focus. One of my very distinguished mentors at the Mayo Clinic, he was really the father of pharmacogenomics, Dr. Richard Wenshibao. He really inspired me with his lectures in pharmacology, saying that there's a large variations among all of us in drug response phenotypes. So we not only study the basic mechanisms of medications, but also the genetics can play a role, and each of us is going to be part of that. So I think genetics is defined as the inheritance in our health condition. But pharmacogenomics is really the places that genetics can impact each of us. So I think that's the motivation for me to start my research as a graduate student on pharmacogenomics. And later, when we published one paper in 2015, when I was a fellow at Mayo Clinic for clinical molecular genetics, that study is one of the most cited reference for the impact of pharmacogenomics. Basically, we studied over 1,000 individuals. They are healthy individuals, but they are at the age of, in the next 10 years, each of the individuals is going to be on multiple medications. I think that study really showed that among all of us, the general population, only 1% of the individuals in that study has no actionable pharmacogenomics. The interesting part, we only studied 5 drug gene pairs in that particular study, and almost everybody, over 99% of that study population, have an actionable pharmacogenomic variant. So I think the bottom line is there are many genetic diseases, there are many genetic testing out there may not impact of us. However, each of us, including you, myself, is taking medication at some point. Each of us, particularly when we're getting older. So I think that's the biggest motivation for me to begin my professional journey on pharmacogenomics. Now I'm in the laboratory side, clinical, to do the pharmacogenomic test. My goal is to advocate for pharmacogenomics so many more people will be aware of the importance of this very special niche area in the clinical lab medicine.
Okay, so you're making the case for the importance of pharmacogenomics in clinical care. Let's just continue down that a little bit. If you think about the state of the field as it exists today, the laboratory tests that are available today, the genes and the science that we know about, if those tests were used in all of the patients where they could potentially be beneficial, what would the impact of that be versus the current state of affairs?
That's a good question. I think if you look at the CDC most updated website information, there are 1.3 million emergency visits every year due to adverse drug events. And among all those patients with the ER, 350,000 patients will end up staying in hospital to be treated for their adverse drug events. I think that amount of financial burden to healthcare, to the patients, to their families, as well as a lot of associated debt, that's the social, emotional impact is huge. I mean, I think the dollars amount is over billions of dollars. So if we can prevent drug adverse events by pre-screening patients, that's definitely going to have a huge impact in our healthcare system and for all of us as a patient.
Yeah, I think it's particularly dramatic when you think of drugs where getting the dose right has a huge safety impact on patients. Drugs like Coumadin. And I'd like to come back to warfarin maybe in a few minutes as an example of what the potential is and whether we've gotten there. One thing that strikes me, though, is when you talk about adverse drug events, that's obviously a really important and really expensive area of healthcare. But on the other end of the spectrum, there are all the cases of undertreatment where patients are being underdosed versus what would actually get them into therapeutic range. And my guess is that most of that is probably invisible. And all you notice is the patient doesn't seem to be getting better the way you thought they would be.
I completely agree with you. I think the drug response, as the beginning of the conversation, is really a spectrum of phenotype. So there's a large variation. People can be rapid metabolizer of a particular drug, could be poor metabolizer of metabolizer status on the other end of the spectrum. So adverse drug response is just the extreme situations, people can end up to die. However, undertreatment could also potentially have other consequences, like a delay of treatment. For example, for treatment-resistant major depression, patients could be easily under multiple trials of different medications until they end up with the ones have a better outcome to them. But most of the patients, about 2/3 of them, do not respond to any of those kind of medications. It's a trial and errors. I think there's a long way for psychiatrists, for neuroscience to be developed, understand fully how those antidepressants work in the brain. But we do know that a lot of antidepressants are within the categories of medications that could have a pharmacogenomics impact. For example, some of the antidepressants like SSRIs is metabolism by cytochrome B450, for example, to C19. And if a patient is end up with a poor metabolizer or intermediate metabolizers, those SSRIs could not work for them well. So the prolonged treatment at the beginning with that particular antidepressant is not going to work for those patients. And the interesting part, I had my 23andMe tested, is a direct-to-consumer type of genetic testing. Turned out to be I'm intermetabolizer for CYP to C19, which means that I have a decreased activity for this particular CYP to C19 enzymes. And if I'm under antidepressant treatment, SSRIs, I could undertreat it. That's one of the phenotype, or I could have adverse events if the dose is beyond that what I can hold. So it's very common. And I think a lot of antidepressant is just one class of medications that really had a lot of attention and had a lot of work in the last 10, 20 years of pharmacogenomics. And there are also other domains of medicine, for example, cardiology and antiviral treatment, anti-HIV drugs, and they all have a good example for pharmacogenomics. Oncology treatment, pharmacogenomics impact.
It may be difficult to precisely quantify this since the field is in flux and the science is in flux, but do you have any kind of a sense of sort of what fraction of currently available prescription drugs are potentially dose optimizable through genetics?
That's a great question. I think we can look at how those medications work in the body. So majority of the medications, I think all of them going through two pathways, right, in the body, phase 1 and phase 2 metabolism. So phase 1 is really oxidation, hydroxylase, all those actions. And phase 2 is conjugation. So the end point of those drug metabolisms is to help the drugs become hydrophilic from hydrophobic to hydrophilic, then eventually they will be secreted out of the body easily. And most of the medications in the market is going through phase 1 particular group of enzyme called cytochrome P450. So those enzymes encoded by CYP genes is very polymorphic, means that there's a large variation of enzyme activity in particular among all the individuals. I think if you look at that point, say, hey, if the medication is going through this pathway, regardless it is engineered as produced by any of the pharmaceutical company, will go through those pathways in the body. And that pathway inherently have a large variation among all of us, which means that the majority of medications could be benefited from pharmacogenomic profiling. Some enzymes are more polymorphic than others, but in general, everyone's liver has those enzymes and the medications go through those pathways.
Really, you're talking personalized medicine, right? Do you use that term a lot?
I think that's a pretty sexy word to be used, personalized medicine, individualized medicine. But by the end of the day, I think each of us has our unique genetic profiles can help us to make a diagnosis and to prevent certain events like adverse drug events. So I would say I like a genomic medicine. I think genomic medicine is at the front end of a lot of medical disciplines. So let's use the genomics medicine as an example, and the pharmacogenomics is really a very practical applications in the genomic medicine.
Okay, so what will it take to achieve this? How would you describe the state of the clinical science in associating different polymorphisms with different doses to make this really practical for clinicians? We're partway there, certainly, but how would you characterize the state of the literature on this?
This field is in general pretty new compared to other domains of medicine. If you look at the history, the word pharmacogenetics was first termed or first coined by Dr. Vogel in 1959. So if we calculate, it's only 65 years. So the field is really new. And there are a lot of literature out there, first beginning with the clinical observation. Now, later, you can resequencing the gene. Now we can actually proactively sequencing the gene to find the drug response related genetics and alterations. But I think there's limited evidence for each of the medication, each of the drug gene pair out there. So there is a long way for us to fully understand every single medication, every single gene, their impact. However, just utilize what we already known for the last 65 years will prevent a lot of bad outcomes out there. And there are examples, very tragic examples. I don't know if many of the audience may already heard about the tragic death of Dr. Kapoor. He's a very well-known Canadian doctor and hospital department chair. And he died tragically in late February because of his treatment related to his stage 4 colon cancer. The doctors concluded after his death that he wasn't killed by his cancer, but was killed by his treatment. So I think if you look at the whole sad story, and it wasn't because of Dr. Kapoor was not screened for his pharmacogenomic gene, for which is the DPYD gene responsible for his cancer medications. Actually, he was screened and the result was negative. The screen was missing one of the variants occur in his genetic profile, which means that the science is out there. We know the gene is important. The DPYD deficiency is important for patients going through fluorouracil treatment. However, the science wasn't there to say every patient may have a slightly different genetic composition. And the common practice for DPYD screen is only focused on the variants already well studied in North American populations, white Caucasians, not in other populations. I think, this is a very revolutionary, evolving domain of science, and the clinicians and the laboratory medicines, including all of us, and the patients themselves need to have a realistic view of this particular domain, saying, yes, we are here so much better than 60 years ago, but there are still a lot of things we haven't really addressed yet. So I think using these tragic examples really demonstrate that education is a critical part of the whole puzzle piece. Yes, we have sufficient amount of evidence out there, and how we can educate the community, the stakeholders, is pretty critical. And the stakeholders also include payers, who's going to pay for them. So there are many roadblocks for us to get there.
Yeah. And I think this is a really important point that you're making, that when we're talking about genetics, to have a really robust evidence base, you have to do pretty large scale clinical studies across diverse populations. If you just do a study in one area of people whose ancestry comes from one part of the world, you're unlikely to get a really balanced picture. Now, when we're talking about drugs, the source of a huge amount of the practical evidence base or prescribing drugs comes from the clinical trials that pharmaceutical companies perform in order to get FDA approval. Obviously, those studies serve a purpose in deciding whether the drug is safe and effective. That's the FDA's mandate. But also, they generate a lot of evidence about side effects, about dosing, about other things. Is the FDA, to your knowledge, moving toward requiring genetic profiling broadly in drug approvals? Are people talking about that? Should they be, if they're not?
I won't speak for FDA for sure, but we all know that medications developed in pharmaceutical industry is one size for all. When all those clinical trials, phase 1, 2, 3, were performed, they were really designed for certain populations, certain age, adults or pediatric population, or patients with certain conditions. But in reality, in real life, the medication impact could be impacted by genetics. Even within that domain of all qualified study participants, they may not be in the same genetic makeup. However, they meet certain criteria to be eligible for those clinical trials. It's very costly if you really conduct a pharmacogenomic studies. And the pharmaceutical companies, I'm not working for them, but we all know if you narrow down a certain medications really to a smaller population rather than a large market, ethnic market, that is going to limit their profit, going to limit their market share. So there's a lot of considerations from pharmaceutical companies. So when the drug can only work for small populations, it's not going to be really big hit. I won't say they require pharmacogenomics informations, but we do know a lot of FDA's drug labeling nowadays include pharmacogenomics indications. Like the example we talk about DPYD. So FDA recently updated their label information to strengthen the importance of pharmacogenomics indication of DPYD in correlation with the urushiol toxicity. So FDA is taking actions, but there are so many drugs out there already in the market. It's going to be challenging to retrospectively conduct the pharmacogenomic studies, and it's super expensive to conduct the full-armed clinical trials to get that evidence.
That is a really good point, that drugs stay on the market a really long time on average. And if a drug's already approved, no one's going to want to go back and spend the money for all of the studies on those drugs. So that definitely is a challenge. Let's come back to payers, because you mentioned them as a stakeholder. How do you think insurance companies look at this area of testing? Do you think insurance companies just look at it like, here's another big area of potential cost? Or do you think that insurance companies are looking at the potential benefits as well?
I think we need to, for this particular aspect, we need to really divide the indication for pharmacogenomics testing, right? So there could be a proactive testing, preventive, screening, preemptive, whatever you say, is when the medications hasn't been administered to the patient, even the patient isn't in that health condition, need that medication, the pharmacogenomics testing is considered as preemptive. So I think for that indications, the screening nature really prevented the payer saying, why I should pay you if you won't get the fluorouracil until this patient's in late stage of colon cancers. There is the long distance of that utility to be fulfilled. So I think from payers' standpoint, say that we need to put dollars for other indications. But the other indications for pharmacogenomic testing to be conducted for patient is reactive, means the patient is already actively being considered for this particular medication or group of medication. And we need to know, for example, the antiviral HIV testing rate. So if patient is already going to go to that protocol, the HIV testing need to be done and is required by FDA. So I think there are two arms of indications for pharmacogenomic testing to be utilized, and the payers are more inclined to pay for the reactive testing rather than preemptive testing. But go back to Dr. Kapoor, we know he's going to under that treatment of fluorouracil, so he's going through the screen, but the screen isn't complete to include his particular genetic variant in DPYD genes. But for others, if you're talking about newborn being screened, each of us, when we were born, all screened with pharmacogenomics profiling, with the current amount of healthcare burden dollars and insurance concerns, is not going to happen. And we talk about the science is the evolutionary process that by the time we are age 55 above, and all the science could be advanced so much than when we were born years ago. So that preemptive testing as a screening tool early on may not be that useful. I think that's a fine balance about where we want to put effort over there.
So let's talk for a minute about the clinical domain here. So if we imagine a doctor writing a prescription on a patient, today, the number of hoops that busy clinician might have to jump through to self-educate on testings available and how to adjust the dose and everything else could potentially be a barrier, I could imagine. So what do you see as sort of the clinical infrastructure required to make pharmacogenomics routine, but more importantly, useful in clinical practice?
I think this is a very important aspect for the whole picture of a clinical implementation of pharmacogenomics in real life. Because as you said, physicians are very busy and it's going to be very hard even read the lab report if that includes information about all the indicated medication related to the pharmacogenomic finding for a particular patient. The more practical tool could be clinical decision support tool built in the EMR. If the patient, for example, like myself, has 2C19, STAR2 intermediate metabolizer status, what medications indicated for that genetic results. Whenever that medication is on my chart, the physicians can be alerted to say, hey, for this particular patient, they need to have alternative antidepressant, for example, or clopidogrel is not going to work for her if she just had heart surgery. I think that will be more practical, helpful, time-saving for physicians rather than let's educate everybody. Yes, the education part is important, but in practical, even you have all the knowledge about all the medication, all the gene drug pairs, it's going to be very hard to do this on an instant setting when the patient's medication is just in front of your eyes. I think it's a challenge. And some institutions, like where I was from, the Mayo Clinic, they do have institutional solutions as clinical decision support tools. It's going to be challenge. It's money. By the end of the day, it costs a lot of investment. But down the line, if you think about all the ER visits, millions of ER visits every year, and if we talk about billions of dollars and related to drug adverse events annually just for America, it's going to be cost effective. But most of the institutions or most of them as a whole, healthcare or payers, they don't really think that way. They have their own institutionally financial goals or other goals. I think this is not on the priority list for many. But that's why we don't see widespread implementation. A lot of folks are working towards that goal. It's getting there. It's making progress.
When I've tried to think about other areas of pharmacotherapy which require personalization, one that has come to mind is weight-based dosing. So you certainly see this a lot in pediatrics. You also see it in adult chemotherapy sometimes. But my understanding is that building that into online prescribing gets pretty complicated. And there have been errors that have come up because of poor user interfaces for weight-based dosing. If you put it in as kilograms when it was supposed to be pounds or vice versa, or grams versus pounds would be an even worse problem. So it brings more variables into it. And there have been patients harmed by this. So it seems like the tools that doctors use, particularly the online tools, like you say, decision support, do have quite a ways to go to hit their potential.
Completely. I think weight measurement is so much easier and cheaper, almost costs nothing, right? And even there's a human error involved, it's a still pretty straightforward measurement.
Exactly. So if it's hard to get weight-based dosing correct in these online tools, it's many times more complicated to get the genetics-based dosing, especially when the science is still evolving and keeping track of all the variants and everything else. I can imagine that could be a challenge.
It's a challenge. And there's another barrier I think we don't talk a lot about, and physicians in particular, when they order genetic testing, there's an important aspect that hasn't been really addressed is the large variation among pharmacogenomic tests offered by different laboratories. So Lab A offers CYP to C19 testing on only STAR2, STAR3 alleles. Lab B offers
Similar test.
Similar test, exactly. So include the STAR2, STAR3, STAR17. Just imagine the same patients being tested by two different labs, A and B will get different results, right? So I think that difference wasn't fully aware by the clinicians. So that's why one professional organization that I'm very fortunate to be associated is the Association of Molecular Pathology and Pharmacogenomics Working Group. This is a group of colleagues, really stakeholders from several professional organizations, including AMP, trying to address this variations in terms of testing build, in terms of testing content, saying we need to have a minimum defined list of pharmacogenomics alleles varied in the same genes that every lab is testing, that those minimum alleles need to include all the well-characterized, well-defined VIP alleles in the genes and being tested across the laboratories. So now back to the physician, when you click the order, you know you're ordering the test that include all of those important pharmacogenomics alleles. You're not missing anything important. Of course, there will be ethnic group-specific alleles that may be not present at the moment, or because of the patient's ethnicities could require additional testing, but at least the minimum, it need to be part of the pictures and then need to come back to the physician in terms of education. You know, there's a whole correlation about from testing, and then we're talking about clinical supporting tools of what alleles and physicians need to take actions upon based on the results from the genomic tests.
So let's go back to this idea of direct-to-consumer testing, patients actually obtaining their own testing. And you mentioned 23andMe a few minutes ago, which is a really interesting example, considering that 23andMe was this huge, high-flying, multi-billion-dollar valuation company until pretty recently when their valuation's gone to essentially zero. So the business model of 23andMe ended up not working out. Do you see potential in direct-to-consumer testing, and what would it take to make that really work out well?
Talking about the direct-to-consumer experience, as I mentioned earlier in our conversation, that we had a genetic testing using 23andMe, and that was actually a science fair study conducted by my two teenager kids. At that time, my daughter wanted to have a project, and we said, okay, let's do that. And also based on the curiosity of my teenagers about their genetic makeup. So I have to order this expensive, like $200 without discount kit for each of us, 3 generations, including my mom living with us in our house. She's over 70 years old. So I said, that's an interesting, very small subject study. Well, however, it's a multi-generational study, right? So my daughter conducted this study. We all have our tests done. We even measured the turnaround time. Some of our kids sent early on, come back sooner. And I think my mom took like 2 months for her to get her results back. The interesting question we were asking for that particular study was, is there any predictive values for this outcome of the study? And as a clinical molecular genetics myself, how I'm going to comprehend these results in the background for my knowledge. It turned out to be, it's very interesting that my daughter even presented this as an outcome at American College of Molecular Genetics and Genomics annual meeting in 2023 last year in Salt Lake City. And she presented and got a lot of audience questions. It's very interesting to see my mom as a 72-year-old woman never have any of the complications except some chronic disease in that age population. Her genetic results wasn't really from 23andMe correlate what we are seeing in her health history. However, both of my kids are very excited about it because they find out who they are looking at the map of, hey, you are 99% of the Chinese population, which is very predicted by looking at them. For myself, I think there are 40 health-related conditions reported in my report, and this particular pharmacogenomics finding on CYP2C19 really making me wonder, so what I'm going to do with my genotype next time I go to the doctors. Is the doctor going to respect these findings? So I think depends on who you are taking those direct to consumers. You may pose different type of questions to follow up your own genetic disease. I have a lot of doubt about what hasn't been included in each of the health conditions tested by 23andMe. For my two kids, they are overexcited about their perfect negative screen results. While my mom is completely mysterious about what's the utility for her even to get tested as a 72-year-old. If she was tested 20 or 50 years ago, is this result going to help predict to prevent certain things happen? So the utility is very variable based on our family experience. However, if we want to get this right in the future, I think the designs need to come from particular laboratories like us, the practice medicines that we follow closely about all the compliance regulatories. We make sure that all the important pharmacogenomics alleles are included, and we make sure the report is going to be very clear about what are findings and what are the limitations. I think the patients need to understand the limitations of each of the tests. And if we can do better in the future, for example, if our laboratories have direct-to-consumer-facing pharmacogenomic testing, and then if I order this as a patient myself, and I can understand better of the results and how to take actions. So there is a long way there, and I think some laboratories are already trying to approved laboratories are already trying to direct-to-consumer type of testing. It's getting there, and then the physicians need to take actions from there and how to incorporate that information when patients present their own pharmacogenomic profiles. Just like if I go to pharmacy, pick up statin for my mom, saying my mom has a SLC1B1 allele, and is the pharmacist going to decrease or increase dose for my mom's statins? Not at the moment, at least, but we will getting there someday, I hope.
One thing you're pointing out is the importance of test design. It really felt to me like 23andMe started off in this sort of Silicon Valley way of let's just do something interesting in genetics and then see what kind of business model we can create out of it, which is very different from starting with specific medical questions and saying, okay, what kind of panels do we need to design to answer these questions properly? It felt to me like maybe they did it backwards, but it also comes back to the work that you're talking about with ACMG, making sure that laboratories have clinically useful offerings.
I think for 23andMe, I don't completely discount their values. I think they definitely play an important role to educate the population the importance of genomic testing and to excite people. We got excited, particularly like my teen kids, they are in this age to be curious about almost everything from universe to their own genes. If they know the importance of genetic profiles early on in their life, they may be more cautious about, be mindful about, or maybe more accepted when they are in medicine as practicing doctors in the future or as a patients. They can ask the right questions. I think 23andMe definitely pose a very important value over there. And also, they shine the light to the traditional lab medicine that we are facing the doctors, we are facing even at the reference lab that there's missing piece over there in the whole puzzle, in the whole conversation, that if we can bring the gap between patients, labs, and physicians, and we can provide a better service in the future. So definitely there's the educational value over there, despite the business model. But moving forward, when everyone could have access to their medical record on their phone, and I think when you have this pharmacogenomics profiling information identified or tested by legit laboratories, it's going to have a much better values for all of us since medications are out there and we're taking all the time for common flu to cancer treatment. So it's going to be very important for the future of our labs.
So just in this past week, the Food and Drug Administration released a final rule for laboratory developed tests. We all saw this coming in the laboratory industry. They'd announced this months ago what they were planning to do, but the final rule just came out. And without getting into the specifics, it appears that the FDA is moving forward with more aggressive regulation of laboratory developed tests. The first question here is, of all of the pharmacogenomic tests out there, do you have a sense of how many of them are currently laboratory developed tests? And any thoughts on how they might be impacted by this new FDA rule?
So first of all, this is very timely and relevant to each of us practice clinical lab medicines. And the FDA move dispatches so much effort. We're trying to push back. The lab medicine community is trying to push back. They insist to move forward, which is very sad move for all of us, including patients. From what I can say, everything on our menu, there are some labs, very few. Surprisingly, 23andMe pharmacogenomic testing is the FDA-approved test. I won't argue about their test content. There's a lot to say about what they included for their FDA-approved version of the direct-to-consumer test. Back to our LDT discussion, that every single test we offer here at ARUP is a laboratory-developed test, which means that all of them is going to under the new ruling of FDA. Also means that we are going to go through the process of FDA clear for all of those LDTs. Thinking about before this FDA rule, we already experienced a lot of roadblocks to really push pharmacogenomics to become a very important tool as part of our genomics health, genomics medicine. Now you're adding all the burdens associated with FDA new regulations, the financial burdens, the regulatory burdens, a lot of the logistic burdens is going to further slow down the development of pharmacogenomics in terms of its implementation. And coming back to the laboratories, we need to decide where the dollars and our energies to put to focus. And of course, FDA is going to be our highest priorities among all the other things. So it's a balance. If FDA asks us to do this, we have to slow down other progresses and further refine our testing and trying to put into educations in advocating to shift it to the FDA-related activities. So it's going to be hindered and slower down, and there will continue to be tragic stories you will see in the news because of lack of pharmacogenomic testing, which could have been prevented if patients is getting those important testing done before their treatment. So I think it's not good news. It is sad in many reasons, but we just continue to do the best we can in this new environment.
In the off chance that any senior FDA decision makers might listen to this podcast, we can always hope, certainly. Do you have any thoughts you would like to share about how you would hope that the FDA would design its process in the future to have more positive patient impact and not so much negative patient impact?
My thought on this is this is a discussion need to be on the same table with all stakeholders. The stakeholders, including FDA themselves, of course, they are regulatory bodies and need to have the laboratory medicine representatives, need to have a pharmaceutical companies also need to be part of the conversation, and the patients, particularly those patients being through the journey that have a very sad tragedies that happen in their families. They need to present all those side of the story together when FDA conceive and process and the proposed plan for any of the actions. I think they did give a laboratory community a window to respond to their proposed rule. However, that window is teeny tiny, that even you have thousands and comments submitted within that teeny tiny windows from individual to a large body of laboratory medicine organizations is not being processed, incorporated in the new ruling. in the future, hopefully, we're all going to be part of that decisions and helping the FDA shape for the better of the future of the lab medicine.
Well, we can try to be optimistic. I love your optimism. Yuan Ji, thank you so much for being on LabMind today.
Yeah, thank you for having me here. It's a really great pleasure to talk to you.
LabMind is sponsored by ARUP Laboratories, a not-for-profit enterprise of the University of Utah and its Department of Pathology. Our producer is Cherie Peterson, with audio engineering by Isaac Acosta Alvarez. You can also find other LabMind episodes at arup.utah.edu, along with an extensive video lecture library providing free CME and CE credits for medical and laboratory professionals. Subscribe to LabMind on Apple Podcasts, Spotify, or your other favorite podcast app. And please leave a rating and review in order to help others find the podcast.